And so just by a show of hands, how many people here are familiar with gene therapy and AAV? Excellent. Okay, so a large number of you, it's a very exciting field, and there are a lot of new trends and new technology. So this session is based on new technologies in a respective field, and today I'm very excited to present some really interesting work that comes from Dr. Luc Vandenberg's laboratory that I worked on with respect to improving upon current therapies in the gene therapy world, and we'll be talking about a specific application in the eye and the ear. So since most of us know what a gene therapy is, I'll go through this rather quickly just to kind of get us all on the same page. In very general and basic terms, if you're looking at a gene replacement or a gene augmentation approach, you're dealing with a cell that is non-functional because of a particular mutation, and as a result, a lot of the times these cells end up dying. However, we can provide the functional gene by a delivery system to enhance its function and allow it to function as closely to normal as possible. And we do see in vivo settings of functional recovery. And the way that we can get that functioning gene into the cell is by a particular delivery system. I have a little truck here, and that is our adeno-associated virus. And so as you know, it's not like most viruses that will get into a cell, replicate, and ultimately lyse the cell. Instead, this is a recombinant AAV, so I'll explain a little bit about that in our next slide. But ultimately, its genome remains episomal, and so it does not integrate into the host genome. And as a result of this, we're able to have gene expression, and because the replication genes have effectively been taken out of that virus, that capsid protein, we do not have replication in vivo. And so a wild-type AAV has a rep and a cap gene between these inverted terminal repeats that are kind of this little loop section on either side. However, when we make AAV in the laboratory or in industry, we don't want that rep gene in between those ITRs because we don't want it to replicate, as I mentioned, in vivo. So instead, you put your transgene of interest. So that gene that you ultimately want to deliver to those cells goes in between the ITRs. And I'm going to be showing you a lot of work where you see very nice green pictures. So here I'm showing GFP as the transgene of interest. And in order to actually produce your AAV, we still need to have replication in our culture dishes so we can make enough capsid for our in vivo application. So you provide the rep and cap genes in a different plasmid. And AAV on itself is what I like to call kind of a lazy virus. It doesn't like to replicate on its own, so you also provide some adenovirus helper genes to allow the production of your specific AAV serotype. And there is a little bit of a menu or different flavors of AAV serotypes. And they are denoted by the rep and cap, as you can see here. So Luxturna, a product that I'll describe really quickly in a couple slides, leverages AAV2. And there is a number of other serotypes, the conventional serotypes really ranging from 1 to 9. And overall, we see using different types of these vectors, very high transduction and infectivity in certain tissues. And so AAV2 has very nice transduction in the retinal pigmented epithelium, number 9 and 5 in your photoreceptor cells. So it seems like the various serotypes have different specificity for different tissues. As I mentioned, its genome remains episomal, and there is long-term and robust expression over 10 years using these types of capsids. Furthermore, depending on the serotype, again, so this is very specific, you can generate high enough titers for certain tissues, especially if you're treating the eye. There are 2 now that are FDA approved, one for retinitis pigmentosa, the LCA model, in addition to spinal muscular atrophy. And one of the negative parts about an AAV is that it does have that limited packaging capacity. So here we have our transgene GFP. It's much smaller than 4.7 kbs, but ultimately that's all you can package in an AAV. That also includes your promoter and your poly-A tail and what other sequences you need within that transgene. So since we're focusing on the eye and the ear today, I'll be describing two different ways to actually target the retina. And one is a subretinal injection, and the other is an intravitreal injection. So the subretinal injection is much more invasive. You actually end up having a transient detachment of your neural retina from the underlying retinal pigmented epithelial layer, because you really are targeting that outer retina where the photoreceptors lie. And then we have the intravitreal injection, which is a lot less invasive. I'm sure a lot of you have heard of the various antibodies, such as Avastin, that help with AMD, and this is an injection that's performed routinely and consistently in an ophthalmologist's office. Not with AAV, though, so this would still require an OR likely, because you are dealing with virus, but it's a very simple approach. You just go through the limbus, and ultimately you're putting your vector in the posterior chamber so that it can now target the inner retina. That's your ganglion cells. So the focus here isn't necessarily the photoreceptor cells, which it is with a subretinal injection, because you can only target that inner retina. And so I mentioned Luxturna. This has certainly paved the way for a number of other retinal applications using gene therapy. If you go to clinicaltrials.gov, you'll see that most of these gene therapies have started with a retinal phenotype or a retinal application. Here I have Lancelot, the first one of the first dogs with retinitis pigmentosa that was treated with gene therapy, and I believe in 2008, around that time when the big paper showing that these dogs did have visual improvement in a various study where they had to go through different obstacles under different conditions of light. Those dogs were ultimately also brought to Congress in the USA to show the promise of these gene therapies. And now we have an improved product. But as scientists, we're always wondering, how can we continue to improve on this? So this is revolutionary. We have an AAV2. What else can we do with the capsid? And so one application, instead of just doing a gene replacement therapy, so you've identified the gene that is missing or due to its mutation, and as a result you just deliver that gene back into the system. Well, there's another application where you can deliver anything that you want. So what about delivering something like ZIAP, which is an anti-apoptotic gene. So it's the X-linked inhibitor of apoptosis. And so in various models of degeneration, you have a very pan expression due to the detachment, for example, of the retina from the underlying neural epithelium, and as a result, the photoreceptors die. So what we did is we packaged ZIAP in a vector, AAV, and we performed a subretinal injection in a number of cats. And as you can see on your right-hand side here, looking at our GFP control, we had very long areas of photoreceptor degeneration and apoptosis. As seen here, the outer nuclear layer is significantly thinner. And then in our ZIAP-treated animals, these were the only two places in the entire sectioned eye where we found a little bit smaller areas of outer nuclear layer thinning. And then two animals, we didn't identify any ONL thinning. And then a third animal, there were some complications from the surgery itself. And as you can see, there is a bit of inflammation in this retina. And so this is what it looked like structurally. We also, of course, looked at the function of these photoreceptors and find that we're not only preventing them from dying, but they're also recovering functionally. And so that is another application for conventional serotypes, but what about designing novel capsids? This is a very big area in AAV research at the moment, and I've kind of listed a number of ways in which people are doing that with rational design and looking at different capsids that are already available in other models, such as the rhesus monkey. But there's also a process shown at the very bottom called in silico reconstruction. And this is when you use various computer algorithms to kind of understand current serotypes as well as those found in other models and try to come up with sequences that may inform an ancestor to the vectors that we currently know right now as those conventional serotypes. And so using this, again, it's all computer-based, so we weren't sure if it was going to work and what exactly we were getting out of these screens, but one vector in particular called ANK80 was produced, and this is a putative ancestor of AAV 1, 2, and 8. And we wanted to explore that further. So we looked at ANK80 itself, and then that little green area around the red dot, which is called its node, and that just means different variants within that node where we induced a couple residue mutations. So the first thing we did was an intravitreal injection, because naturally we want to be able to improve the transduction after an intravitreal injection with different capsids. So AAV2 does a good job after IVT. However, the transduction is still very low, especially when you move to larger animals. So when we inject AAV2 in the rhesus monkey, we see that you really only are targeting the very inner retina, that nerve fiber layer and the retinal ganglion cells. And we thought, maybe these in silico reconstructed vectors could do a better job. Here we go. They did not. So we had very low expression right around the optic nerve. That's fine. Let's try other models, and let's try other types of injections. So we did a subretinal injection, and we saw right away this is a fundus image. So we basically hold the camera up to the eye of the mouse, and we look at the fluorescence. Again, our transgene here is GFP, and we saw robust expression of our transgene, and we're very interested to see what that looked like in our retinal sections. So we compared it to AAV2, AAV8, and then the different variants of ANK80. And we found that ANK80 elicited robust expression in those photoreceptor cells, as well as the retinal pigmented epithelium and the Mueller cells, as you can see here. So those are the support cells that kind of span the entire retina. So this was intriguing. Since we're discussing preclinical work today, we naturally moved into a larger animal. So here we're looking at the non-human primate. This is a monkey model. And we injected AAV9, AAV5, and our ANK80. And as you can see, there is variation, and we're not afraid to show that. So at the very bottom, you'll see our ANK80 variant L65, which we found is the most promising candidate in the mouse studies, and that's what we wanted to inject in the monkey. One animal we had lower transduction, and the other one we had higher, and as we continued with further experiments, we also saw a robust expression, but this is just to show you the gradient there. And indeed, we saw that we were able to target those photoreceptor cells. And so this is really informative to future applications, because you want to be able to target as many of those photoreceptors as possible to elicit that gene replacement or augmentation therapy to improve the disease phenotype. We also found, showing at the very far right, that we could transduce both the rods and the cones. And so we moved this naturally into a mouse model of degeneration. You have, I'm sorry it's difficult to see, but on the right-hand side we have our uninjected or knockout control, and here we have our ANK80 replacement, and we have a very nice structural protection in addition to proper localization of the gene that we were replacing. And so this was naturally also followed up with functional tests, and we saw a very nice functional improvement as well. And this is quite promising and is currently being developed as a therapeutic in humans. And so let's try another organ. So we wanted to see, how does ANK80 do in the ear? This is just to kind of give you a little bit of background on other serotypes. So AAV1 at the time was really the gold standard, and it did a great job of transducing the inner hair cells. However, it's the outer hair cells that you want to target as well, because they will modulate and tune the noise. And so you really need to get your gene replacement therapy into both sets of cells to have functional improvement. We also compared this with AAV2 and AAV8, And when we injected our ANK80, we found that there was transgene expression of both the inner and the outer hair cells. Sorry, I'll go back. And this is a really great paper by Lucas Landiger, 2017. I recommend you take a quick look. It's really neat that we had such high expression, up to 90% in some of the mice using ANK80. And naturally, what is this application? So you can use this vector to correct a model of Usher syndrome. So here we have a model where there is a mutation in the harmonin gene, which causes a very severe retinal phenotype. In patients with Ushers, they also have the eye and the ear and hearing loss as well. So it's a devastating disease affecting both organs. And so this work was primarily centered around the ear, and a round window injection was performed to deliver that harmonin gene that is mutated in this mouse model. And so here we have the signal for when you have your mutant mouse, as well as a mouse injected with harmonin. But when we provide our ANK80, either the harmonin isoform A or B, you can see that we start to get hearing back in these mice. And so this was a DPOAE model where you place little electrodes into the ear and you're able to have a readout of potential decibel hearing from that mouse. And the blue down here shows the mice who are the knockout mice, and they are unable to detect anything along that scale of hearing with respect to the decibels. The black part at the very bottom is the wild-type animals, so a mouse with normal hearing, and here we have our gene therapy-treated mice. And you can see that overall there was improvement up here when we pulled all the mice together, and a couple of mice specifically had very strong response to this gene therapy intervention, and their hearing was was almost similar to that of a wild-type animal. So, in conclusion, there are many ways to innovate new gene therapies, which include capsid design and also what you're packaging into that capsid. You can use in silico reconstruction, rational design, discovering those naturally occurring vectors, and today I presented to you some work on in silico reconstruction. With respect to preclinical studies, so a lot of the validation with respect to tropism, as I've shown you, is performed in mice. However, it's always a great idea to go to larger animal models, such as the non-human primate, which I showed you, as well as the feline. There's also a number of dog knockout animal models that are available to test various gene therapies, especially in the retina. A lot of those are out of UPenn, and also to test your proof of concept. And with the recent FDA approval of these types of gene therapies, and hopefully very soon Health Canada, it's likely that we'll continue to see more in the pipeline and have these therapies available for us. But let's design them ourselves. So again, we're interested in new technologies. If you're designing capsids or have a really neat gene therapy idea, come and see CCRM, and we really look forward to working with you. Thank you very much.